I think this topic will follow pretty well from what we just heard, actually. I'm going to talk specifically about how Gould's ideas have influenced primarily my thinking about genome evolution, and I think it will tie in with a number of the topics we've heard about so far today. So, in thinking about how to organize this, I thought it might make sense to come up with a short list of lessons that I have taken from reading material produced by Professor Gould. And I think one of them that's pretty interesting and important and has already come up today is this notion of narrative. So the idea that the details of what Dr. Lewontin called pure history are important. So knowing the specifics of how something has changed or how some lineage has evolved, but also how somebody's career changes with different events, I think is important. And Gould very much emphasized that. The question of origins, and more specifically distinguishing origins from things like maintenance or persistence, origins from function, these things can be decoupled. So the reason that a trait evolves in the first place may not be the same reason that it's still around today. What it was doing initially may not be what it's doing now, if anything. The notion of exaptation has come up also. The idea that a feature that is adaptive for some other function or has no function at all can acquire one. It can be co-opted to serve a new function. The importance of development and drawing connections between what goes on at the genetic level and what you see at the phenotypic level, that's a very important component, and I'll talk about that a little bit too, although not in the same way as we just heard. The notion of pluralism, which is gets into this idea of an expanded synthesis, but basically the idea that small genetic changes that accumulate slowly by natural selection is not all there is in evolutionary theory. So there's three major components there, three major claims: small genetic changes, slow accumulation due to natural selection. All three of those things turn out not to be the entire story. The concept of contingency, namely unique events, and this I would say ties in with the narrative aspect or the pure history aspect. Sometimes there are weird things that happen once and have big consequences, and Gould loved unique examples. The notion of hierarchy or multi-level selective processes, evolution operating at multiple levels, that's quite important in what I'm going to talk about today. And finally, I throw this in, the notion of scholarship, and we just saw a very good example of knowing the history of one's field. If you don't know the history of your field, if you don't read the original literature that led to the current state of the field, you're always going to be surprised by some new discovery. You're going to overhype that discovery, and you're going to see claims that this brand new thing has been said, when in fact it was said in 1972. I'll give you some examples of that as well. Just skipping to this idea of narrative, how did I get interested in genome evolution? Well, I was actually focused primarily on organismal biology as an undergraduate, and the individual who ended up being my PhD advisor gave a seminar, and it was called Frozen Phenotypes, and trying to understand this idea of stasis. And I emailed him some questions because I had been reading Gould and Eldredge and others and said, I've been very interested in this problem, and sent him some questions. And he invited me to come and see his lab. So I did, and we chatted, and he introduced me to the topic I'm going to show you today, and I found it especially interesting because I could see the connections to these questions. And so that contingent, unique event has pretty much shaped my career trajectory, and it was very much influenced by Gould very early on. So what is that topic, and what do I work on for the most part? Well, on the face of it, a very simple question. Here's how Science put it. They had a special issue in 2005 where they listed 125 questions we still need to answer. And there's lots more than that, of course. But one of them was this one. Why are some genomes really big and others quite compact? Okay, seems like a very simple question. They give you an example. Pufferfish genome is about 400 million bases, or 1/10 the size of the human genome. And the lungfish is 133 billion bases. Why the difference? Seems like a simple question. Before I get into why it's not a simple question, let me just remind you what I mean by the term genome. So if we go back, history of our field, what is the original definition of genome? It goes back to Hans Finkler in 1920, and he says the genome, which is thought to be a combination of gene and chromosome, it's the haploid chromosome set, which together with pertinent protoplasm specifies the material foundations of the species. Okay, translation. Under the original definition, it means two things. One, the total amount of DNA that you have in your chromosomes, or what we now call genome size. And number two, the total complement of genes, or things that are specifying development, what we call the genotype. It can't mean both of those things at the same time. I'll show you why. In prokaryotes, bacteria and archaea, there's a strong link between genome size and gene number. That's totally not the case with eukaryotes. When I say genome size, I'm going to be meaning the amount of DNA in one copy of the genome. In the 1940s and early 50s, it was observed that the amount of DNA per genome was relatively constant within a given species. That is to say, all the individuals in a species and all the cells in an individual organism have the same amount or some multiple of that amount. So there's a constancy there. And that led to this term C value, for constant value. The fact that DNA amount is constant in each cell and protein is not was taken as evidence that DNA was the hereditary material and protein wasn't. So that's how early this observation was made. If we look across eukaryotic species, though, there's at least a 200,000-fold difference in genome size between the smallest and largest in eukaryotes. Even in animals, it's about 7,000-fold. That's a pretty big difference. One of the surprising observations that was made early on was that the amount of DNA is not correlated with how complex the organism is, or how many genes you might think it has, for example. So here's the problem. DNA is constant because it's the stuff that genes are made of, but DNA amount doesn't correlate with number of genes. So this became known as the C-value paradox in the 1970s. This is just an illustration of some of the variation. This is based on about 10,000 species. Humans, you can see, humans are up here at the blue star, very average in size for a mammal, but there are lots of groups that are quite constrained in genome size, like birds. Others, like amphibians, are very large in their range. Lungfishes, as you heard, have huge genomes and so on. And it's not linked in any intuitive way with complexity. Now, we know the answer to the C-value paradox. It's that most DNA is not genes. The vast majority of it in eukaryotes is non-coding DNA, what we call them. So this solves the paradox. There is no paradox. You shouldn't expect a large genome to be found in a complex organism. There's no reason to expect that. But it does raise all kinds of other questions, such as, where does this non-coding DNA come from? How is it spread and lost over evolutionary time? Does it have any implications for organism phenotypes? Is it functional in some way? And why do some groups have so much of it and others have so little of it? That famous question that Science asked in 2005. Well, we know a few things about it, and again, going back to very early work, we know that the amount of DNA is closely linked to the size of the cell and also how quickly the cell can divide. So here are some drawings done by George Gulliver in 1875 showing nucleus size and cell size being strongly correlated. Now, there's no way he knew what was in the nucleus, but he did document this relationship. I've since done these photomicrographs. They're all at the same magnification, and they are species with different genome sizes And you can see, for example, in a Siamese fighting fish, small nuclei, small genomes, small cells, whereas in a lungfish, large nuclei, large cells. And frankly, you couldn't fit the nucleus of this lungfish inside the entire cell of that Siamese fighting fish. So at some level, these must be causally linked. So what does this mean for organisms if you've got genome size related to both cell size and cell division? One possibility, if you make every cell bigger and you don't change the number of cells, you'll get a bigger body. And we do have, in some cases, correlations between genome size and body size, mostly in invertebrates. We also have a link between cell size and surface area to volume. So this is influential on gas exchange and metabolic processes. So it could be that genome size is correlated in some way with metabolism. And I mentioned that the other correlation is with cell division rate. And so this could play out as a relationship between genome size and developmental properties of organisms. There's lots of examples in animals and plants and other groups where this is true, but I'm going to highlight a few that I think are especially interesting and might have been especially interesting to Professor Gould for reasons that I hope will become clear. If you look at patterns of genome size in birds, for example, remember they have relatively small genomes across the board. But even within the group, you can see patterns like the flightless birds have the largest genomes and the strong flying birds have the smallest. The hypothesis that was put forward is because flight is so metabolically demanding, they need to have very small cells to fly for purposes of gas exchange, which means there's a constraint on the evolution of genome size in birds. We're not satisfied with just these little categories, though. So we've gone through and looked at genome size versus things like wing loading versus metabolic rate. And we also made a prediction. Surprisingly, nobody had ever measured a genome size in a hummingbird. If you think there's a relationship with metabolism, you would expect hummingbirds to have very small genomes, and they do. They're down here at the smallest range of all birds. Bats also have smaller genomes on average than most mammals, so it looks like flight may in fact be a constraint. But this is where I think Gould would be interested, is the pure history aspect. So we don't know, just from that observation, whether flying vertebrates got smaller genomes, they jettisoned genomic baggage, as I said, or whether they just never had big genomes, they just stayed small. It's difficult to answer a question like that using only observations of living species. So the way you go about it, some colleagues at Harvard have gone about it, is to look at fossils. Genomes don't fossilize, but the spaces in bone that the cells make, the osteocytes make, do fossilize. And you can correlate that against genome size in living species and then infer what genome sizes were in extinct species. So they've gone through and looked not only at different lineages of dinosaurs, but also at pterosaurs and their non-flying relatives, and found that, in fact, genome sizes were already slightly small compared to most other relatives in those lineages. So here's Gould's favorite, the Tyrannosaurus, and you can see that this lineage that is now represented by birds, the avian dinosaurs, had smaller inferred genome sizes than other non-avian dinosaurs at the time. Now, these are not as small as most modern birds, so there was continued reduction after that, but they started out small to begin with. So there's the pure history that I think Gould would be interested in. A brief example about development. There are correlations between genome size and developmental rate in a variety of different organisms, including in insects and amphibians. But where it gets really interesting, I think, is when you look at the complexity of development. So when you have to do a lot of developing or not very much developing and time is limited, for example. And one area you see this is in metamorphosis. So if you imagine the standard biphasic lifestyle of a classic amphibian, a frog, adult, egg, tadpole, juvenile, with a tail, and then back onto land. Very different life cycle stages, very different adult and larval stages. Whereas there are, of course, amphibians that don't have metamorphosis at all. what I've got here, and I'm using very technical terms, so try to follow small genomes, large genomes, and huge genomes. If you look at frogs that live in short-lived pools, like in the desert, very small genomes. They have to come out, do their whole life cycle very quickly, back underground. Then you've got frogs that have normal development, salamanders with either biphasic development or direct development. And then you get into facultative neotenes, which are salamanders that will metamorphose under certain conditions, like the axolotl here. Inducible neotenes that you can cause to metamorphose in the lab. And obligate neotenes like Amphiuma that can't metamorphose. If you inject them with the hormone, they die. This is the order of genomes in that group. So when you lose metamorphosis completely, it seems like you have the growth of a very large genome. And these, again, with fossil data, are thought to be secondary increases. So there's been an expansion of the genome after they lost metamorphosis. You also see this in insects. So on the top are what we call holometabolous orders. These are the insects that have complete metamorphosis, egg, larva, pupa, adult. And on the bottom are the hemimetabolous or ametabolous orders that have no metamorphosis or no complete metamorphosis, egg, and then a series of nymphs that look a little bit like an adult. In this group, which is the best studied, we can't really find many genome sizes larger than about 2 billion base pairs. In this group, where there's no metamorphosis, you can find some genomes that are 3 or 4 times larger than yours. So it looks again like there's a bit of a constraint on genome size based on development. So this raises the question then, what is in a genome? Well, thanks to complete genome sequencing projects, of which there are hundreds now, we have pretty good information for a number of species, including ourselves. This is a breakdown of the human genome. If you check out protein coding genes, that's the exons that encode protein, less than 2% of your DNA encodes proteins. By contrast, probably half or more, I'll show you a new estimate in a second, is what are called transposable elements. These are bits of DNA that are able to take themselves out of their position, make a copy, and reinsert elsewhere, or to amplify by various means, independently of normal replication. So they've been described as parasites of the genome. So 2% are the protein coding regions, and half or more are virus-like elements that live and reproduce in your DNA. I said about 45%. This new estimate suggests it's probably more like 65 or 70% of the DNA, which is not very surprising. And I do want to point out, this was actually alluded to, some of the things that we now know, including this idea of transposable elements, of non-coding DNA, and so on, some things that we have learned since the modern synthesis. DNA is the hereditary material, as we heard. DNA has a double helix structure. DNA and RNA use a triplet-based genetic code. Genome size does not correlate with complexity. Most eukaryotic DNA is non-coding. At the molecular level, most changes are neutral, so they're not adaptive or detrimental. They tend to just be small-scale turnover. We know that there is, or at least was, a lot of transfer across species, especially in bacteria. We know that duplications of individual genes or entire genomes have been very important in evolution. For example, there was a genome duplication around the origin of vertebrates, and another one around the origin of bony fishes that's not shared by other vertebrates. We know that development is regulated in part by what are sometimes called master control genes or developmental regulatory genes. These are the product of gene duplication. We know that many times structures that are analogous at the organism level are homologous in some way at the genetic level. So there are some complexities to those definitions. So in other words, some of the some of the genes that are involved in regulating eye development are the same in mouse and in fly, even though the eyes that they produce are different. We know that morphological similarity and genetic similarity are often decoupled. So for example, archaea and bacteria look similar, but most phylogenies put archaea as closer to eukaryotes, that is, for example, closer to us than to bacteria. And we also know that genetic similarity does not necessarily equal morphological similarity. So humans and chimpanzees are very similar genetically and very different in many important respects. We know that complex organisms, including ourselves, we don't necessarily need a lot of genes. Some estimates were as high as 100,000 genes. It's probably more like 20,000, which is not so much more than a nematode, and it's less than rice. We know that individual genes may encode multiple proteins by a variety of mechanisms, and we know that transposable elements have played an important role in many major transitions in evolution. None of this was included in the modern synthesis. It couldn't be because it wasn't known at the time. On this last point, I just want to bring it up again. This idea that transposable elements have played important roles. One way that has happened is they have taken on a function. They're parasitic initially, and they become functional for the organism, which is very much in line with ideas that Gould had early on. Again, similar to the story with development. He had ideas that have since been borne out. So in 1982, he and Elisabeth Verba published their paper on exaptation and included as one of the examples co-option of selfish DNA, parasitic DNA, transposable elements. So it was right in the original description of that term to use this as an example. They went a little farther. Jürgen Brosius was here this morning. He had to leave, unfortunately. But they published a paper in 1992 that sought to develop an entire nomenclature around these elements, having to do with whether they took on a function or not. That particular nomenclature has not taken off, but the idea of non-coding elements taking on a function is very much part of modern genomic science. Again, this idea of knowing the history of one's field. If you read almost any paper or media report about a non-coding element in the genome that's functional, you will read the following introductory sentence. Once long dismissed as totally non-functional, selfish DNA is now blah, blah, blah. It's almost in every single report, except if you go back to the original papers, so one of the first discussions of junk DNA, being junk doesn't mean it's entirely useless. Common sense suggests it would be discarded. If so, there are several functions for junk DNA. If you go back to the original paper on selfish DNA, it would be surprising if the host genome did not occasionally find some use for particular selfish DNA sequences. One obvious use would be for control purposes at one level or another. This has always been part of the thinking about junk DNA and selfish DNA. And in fact, the Orgel and Crick paper and a Doolittle and Sapienza paper in 1980 were written specifically to counteract the same kind of adaptationist thinking that the Gould and Lewontin paper was about the year before. That is, because it's there, it must be functional. Let's just keep thinking of reasons why you need so much DNA. They were arguing you don't need those reasons because it can be there for some other explanation can account for it, namely that they make copies of themselves and they're good at that. But I do want to give you a couple of examples where this notion of exaptation at the genomic level has turned out to be correct. In the evolution of the vertebrate immune system, for example, what you've got are a series of immunoglobulin genes that are cut and pasted and rearranged in different combinations. This gives you all the diversity of your immune responses. What kinds of elements are good at cutting and pasting DNA? Transposable elements are good at cutting and pasting DNA. So an integral protein in this, what's called the VDJ recombination system in vertebrate immunity, is derived from a transposable element that took on a function. If you look at the evolution of placental mammals as another major event in evolutionary history, you will still, again, find traces of transposable elements having an influence. So, for example, if you are a placental mammal, you've got a foreign body developing inside the mother. It's not genetically identical. The immune system would otherwise attack it. So you need to suppress the maternal immune system. And one way that that is accomplished is via the action of what's called an endogenous retrovirus, also a transposable element. They also play a major role in the development of placental tissue, in the syncytin genes and so on, are also derived from TEs. Another thing that I think Gould would be very interested in terms of modern genomics is this idea of what I sometimes call non-standard genetic mechanisms. So this is not a small-scale mutation. You get a new allele, you get slow change in the population through natural selection. These are otherwise strange mechanisms, or they happen one time and they have large consequences. Developmental regulatory genes is a good example. But here's a few that have to do with human uniqueness, and we'll hear more about humans tomorrow. There's a duplicate copy of a particular gene that's involved in brain development. That gene was inserted, that copy, sorry, of the gene was inserted by a long interspersed nuclear element, which is a type of transposable element, right before the origin of the apes. There's another one that was deactivated in humans that has consequences for brain development and function, and it was deactivated because a particular transposable element inserted into it. And here's a paper that is not even published yet that shows a gene duplication resulting in a slowing down of neural development, which results in the formation of more connections, more complexity in the brain. It's thought to potentially have been important in brain development in humans relative to other primates. So again, this idea that a one-off event, accidental gene duplication, can have these consequences. And the last thing I want to talk about here is Gould's notion of hierarchy. Again, he found some connections. He was obviously a paleontologist, not a molecular biologist, but he found connections between genomic processes and his thinking at other levels. So I particularly like this quote from 1992. "Punctuated equilibrium is but one pathway to the elaboration of hierarchy, and probably not the best or most persuasive. That role will probably fall to our new understanding of the genome and the need for gene-level selection embodied in such ideas as selfish DNA." So the notion there is that these transposable elements evolve and reproduce inside the genome. There's a level of intragenomic selection going on, and that this can then be linked to higher levels, like the organismal phenotype. And it's interesting to me that although I don't think these gentlemen ever read each other's papers, there was another one by a molecular biologist who said more or less the same thing, but coming from the opposite perspective. So Gould was always interested in relative frequency of things. Something might be an important idea, but does it really happen very often in nature? He was always interested in those kinds of questions. And thinking about selfish DNA, he went the same way here. So he said, how much DNA is self-centered DNA? If the answer is lots of it, then we need a fully articulated hierarchical theory of evolution. He also said if not very much is, then who cares? Neat idea, doesn't really matter. Well, okay. Each copy of your genome contains about 20,000 protein coding genes, or about 2% of your DNA, whereas one type of transposable element, called line 1, is present in more than 500,000 copies, and another element, a short nuclear element, called ALU, is present in over 1 million copies. So you've got 20,000 genes and 1 million copies of ALU in every cell in your body when you're walking around. In total, there are about 3 million transposable elements in your DNA. So I did the math on this. I sort of thought, okay, this is about half the human genome. There's 10 to 100 trillion cells per human. There's 7 billion humans on the planet. How much selfish DNA, just in humans, is there walking around? It's about 2 million metric tons of selfish DNA, or 4 times 10 to the 30 copies on the planet, just in humans, and we have a relatively normal-sized genome, not particularly big. So I would say Gould was wrong here. It's not lots of it, it's lots and lots and lots of it. That means, according to this claim, we need to think seriously about multi-level selection because of what's going on in the genome. And that's very much influenced my thinking. So, for example, we have lots of different mechanisms that can result in changes in genome size. Transposable elements is probably the biggest one, but chromosomal mutations, duplications, and smaller scale mechanisms. These influence cell size and cell division, and in some models, they influence cell size via cell division. And then these have cascading effects up the level above, so metabolic rate, body size, organ complexity, which I didn't talk about, developmental rate, and then these can have influences on the ecology of organisms. But that's not really hierarchical, that's more just showing you a connection from the bottom up. So I want to just take a second and think about how hierarchy theory and thinking about multi-level processes can help us understand what otherwise have been mysterious observations about genomes for 60 or 70 years. One possibility is that the genome size of a particular species, that question that science had, why are some so big, why are some small, it could be the balance of different levels at which selection is operating. Intragenomic selection, favoring accumulation of transposable elements, making genomes bigger, constraints on the organism, on their development or metabolism, making genomes smaller, and somewhere in the middle is what you get. It also means that genome size itself may not be adaptive. It may not be sensible to say, oh, here's a genome with 8 billion base pairs in it, why is that optimal for that organism? It may not be. It could be the product of selection, but not be adaptive in its own right. And, of course, this very notion of stasis, or constancy, or C values, the C standing for constant, it could be that the stability of genomes is a result of multi-level selection. It could also be that the spread of these elements is constrained by properties of the organism. So if you have flight or metamorphosis in the desert, you will have a small genome. It will keep those elements in check. On the other hand, if those properties of the organism change, you may have an expansion of those elements. If you lose metamorphosis, if you become flightless, genomes may suddenly start to expand. And that these constraints are not universal. They vary according to the biology of the group. So in birds, it looks like metabolism is important. In amphibians and insects, development is important. So here's a more hierarchical view, and this diagram was from a chapter in a book that I edited. And when I drew it, it was very much influenced by Gould's thinking and by that quote about needing a fully articulated theory of evolution. And this is my version of a hierarchical approach to the question of genome evolution from that perspective of genome size. So you've got selfish DNA, transposable elements, increasing genome size or influencing genome size through intragenomic selection. This has effects on the cell in terms of cell size, cell division rate. This has consequences for organismal biology in terms of body size, metabolism, developmental rate, so on. This can have impacts on ecological lifestyle. If you have a huge genome, you're not likely to be a group that can fly or develop rapidly. But it goes the other way as well. So if something changes at the ecological level, flight is evolving or high metabolisms are evolving, there may be pressures related to metabolism or development which can influence the evolution of traits at the cell level, including cell size and cell division. These will create indirect selective pressures on genome size because of its close link to cell size and cell division, which will create a process of sorting among elements within the genome. So either there will be a lot of deletion or otherwise constraining their evolution at the level of the genome again. So bottom up and top down, I would call that a fully articulated hierarchical understanding, at least of that one question within evolutionary theory. But I think Gould's thinking on this has very much helped us to clarify a number of different points in there that had been debated and really not very fruitfully for a number of decades prior. So with that, if there's time, I can take a question or two. If not, then thank you.